Hello, this is Mark Burow, and today I'd like to present some of the work we're doing to develop new runner peanut varieties, ones combining tolerance to water deficit stress, resistance to root knot nematode, and high oleic seed. Losses from water deficit can be quite high in peanut, estimated to be as much as $500 million per year on a worldwide basis. In the southwestern United States, all peanut acreage is grown under irrigation. The rainfall levels in the West Texas region typically are 14 to 18 inches for the entire year, or less during the growing season. In the eastern New Mexico, West Texas area, the irrigation water is supplied by the Ogallala Aquifer. However, this is declining rapidly, and as a result, many growers are finding it difficult to supply sufficient water for their crops, especially during June, July, and August when evaporative demand is highest. Root knot nematodes can cause significant yield losses in infested fields. Fortunately, there are sources of resistance that are known, all tracing back to the Texac6 or specific amphidiploid that Charles Simpson developed. We do have DNA markers we can use for selection. High oleic oil, we're developing high oleic varieties because these have longer shelf life. Also, they're better for the coronary health of consumers. In this talk, I want to present three general things: the model and screening methods that we're using, markers used for selection, and then results of field evaluation, primarily for yield and replicated trial. What is tolerance to water deficit? The definition I'm using is the ability to sustain profitable yield under reduced water availability. I think, at least for growers, this definition makes sense. We could do screening for yield alone, and some programs do that. The problem is there are many genes associated with yield and high genotype by environmental interaction. Also, we need to select for other things, such as high oleic, grade maturity, and disease resistance. Combining these all together in a single package can be difficult. I prefer a trait-based approach for several reasons. NIGM demonstrated that some environments, including one similar to West Texas, trait-based approach was superior to selection for yield alone. Also, we can measure different response mechanisms of water deficit stress and potentially combine them together. And if we can get DNA markers for these as well as other traits we're looking at, we could do simultaneous selection in a very early generation for all of these. And for this, we use the minicore collection for identification of sources, tolerance, resistance, and markers, which I will mention after I talk about our selection methods. Our screening methods are based on the Burow equation, which defines yield as a function of transpiration efficiency, and harvest index. In our model, we use canopy temperature as a proxy for transpiration, bad chlorophyll readings as a proxy for transpiration efficiency, and harvest index, when we're able to take it, is very tedious, involving bagging the entire plot. We're also doing measurements of flowering. We want something that can continue flowering during water deficit. Periheliotropism, that is, ability to fold the leaves during the heat of the day to reduce evaporation and excess sunlight, and also measurements of plot growth, such as plot width and height. The markers we used for selection came from the peanut minicore. This was evaluated over 2 years in 2 locations in West Texas, and we had 3 water treatments. The drought stress was done between approximately 40 and 105 days after planting, and you can see 3 different levels of water deficit stress. 25% evapotranspiration replacement corresponds to roughly 1.5 to 2 inches of total irrigation and rainfall in a space of a month, June, July, and August. We did biweekly field measurements, and then post-harvest data we took also. For GWAS, we used 438 map microsatellite markers, and based on linkage disequilibrium, estimated this covered 1/3 of the peanut genome. There were a number of gaps, but at least it gave us some initial data. And from the markers, we either used a strict Bonferroni level of significance or less stringent significance, but saying that we would only select the marker if it were present in at least 3 of 12 environments. And across these 9 traits, we had anywhere from 1 to 8 different SSR markers. This is the work of Vikas Belkar for his master's thesis. Marker validation was done in F2 populations as part of the thesis work of Jennifer Chagoya. This was done in 2 different populations. One developed by Michael Baring to combine nematode resistance and high oleic trait with water deficit tolerance. Another one I developed to combine early maturity and medium seed with water deficit tolerance. The markers that were used were the microsatellite markers from the minicore collection for water deficit. A rapid marker developed for root knot nematode resistance, and cast markers developed in our lab for the high oleic trait. Selection was done in the F2 for homozygotes or heterozygotes. Markers were evaluated in two populations. In population 1A, it was shown that 6 markers were associated with yield. Population 2, 3 of the markers could be validated. In the case of population 1, selecting the 3 best markers gave a 15% yield increase compared to the unselected population as a whole. Population 2, the increase was only 2%. This was not statistically significant. Down below, this graph shows an example of one of the markers. The blue bars are cases where we had homozygotes for the favorable allele of the marker. The red were the individuals that were homozygous for the unfavorable allele. You can see the lower yielding material had a higher proportion of the homozygotes for the unfavorable allele. Evaluation of breeding lines was then done in the field. These F2s were carried forward to F3 and succeeding generations, evaluated and replicated yield plots. This was done in 2 locations, Texas A&M AgriLife and the USDA ARS. Texas A&M AgriLife, we had 110 lines. These were generally not selected. We did have the marker selections, but also ones that were not selected. This was done under flood irrigation. USDA ARS, we took 40 lines that were selected with markers for tolerance to water deficit and resistance to nematodes. And in South Texas, the evaluation was done on a commercial farm with the same 40 breeding lines. This slide shows yield differences under water deficit. This was done at Texas A&M AgriLife with 110 entries. The 3 points I want to make. One is there are several accessions that do quite well under all 3 years. And even though the overall means did not show statistically significant differences, the best materials and the best 10% quite frequently showed that these materials did well. And so I think that additional testing will show across years we'll have some entries that can be demonstrably superior to others. Second is if we take the best yielding materials for each year and look at them, quite often they do statistically better than the check varieties here. And across all years, you can see that these best materials yield anywhere 20 to 50% higher than the check varieties. So that shows these materials, some of these are significantly better than check varieties under water deficit. Third point is a number of these materials, those in the blue boldface, were selected with markers. Those in black Roman type were not. And when you look at the mean yields, those selected with markers out-yielded the unselected by 20%. And this corresponds to the similar differences we saw in the F2 generation. At the ARS, several accessions were in the top yield category, and a number of these were also ones, you see highlighted in blue, that did well also at AgriLife. Several of the breeding lines also out-yielded checks, shown in purple, and a few of these accessions also had good combinations of yield and grade, although few of them graded numerically as high as Tamarron 0011, which graded very well, although it did not yield as well under water deficit conditions. Under irrigation, we'd see that yields of the check varieties generally were at the top of the test, although one accession, entry 30, yielded at the top of the test in 2 years numerically, as well as had a grade that was comparable to what we see among checks. A number of the accessions here, highlighted in blue or green, had yields that were near the top of the test under water deficit and did well also under irrigated conditions. Although in general we can see the yield of these under irrigation were numerically lower than what we would see for check varieties. We also see that the grades of a number of these breeding lines numerically were less than what we see for check varieties. Seed sizes of these in general were similar to what we would expect for runners, typically 60 to 75 grams per 100, although a few had seed weights that were smaller. We do a comparison across treatments and locations. What we see is that several of the accessions did well across all environments. Those highlighted in blue, for example, did well both under water deficit, ARS and AgriLife, and in South Texas, even under full irrigation. Again, we saw the differences, breeding lines under water deficit generally did better than check varieties. The opposite was true under full irrigation. So several breeding lines seem to do quite well under all conditions. In comparing across irrigation treatments, we can see several things. One, it appears that we have a case of tolerance to water deficit stress as opposed to enhanced yield potential per se, because those materials that yield the best under water deficit stress, the breeding lines, were not the same as those that yielded the best under full irrigation, which were check varieties. Now, breeding line 30 yielded well under both sets of irrigation conditions. However, although it graded relatively well under full irrigation, its grade was somewhat poor under water deficit stress. There are several other breeding lines that did well for one trait under both irrigation conditions, but did not do so well for another trait. Of major concern in this population is grades, because in general, grades were fairly low under most of these materials compared to check variety Tamronowell 11, for example. And this is probably due to use of an unadapted accession as donor parent. We may need to make selections separately at this stage for irrigated and water deficit conditions, because very few, if any, of the accessions seem to perform ideally under both sets of irrigation treatments. In conclusion, then, for future work is we saw significant associations between SSR markers and phenotypic data in population number 1, the F2 generation, and also in the row plots. We saw several breeding lines yielded well under water deficit with potential for release. Grades were generally low, but may be satisfactory for very few lines. Well, ongoing work, we're doing additional field evaluations to select for good yield and grade. We have another set of breeding materials, which I haven't mentioned here for lack of time, that we're doing similar work on. We're doing marker-assisted selection for homozygotes within those best breeding lines. The idea is we might be able to get some additional improvement. We're running GWAS with SNP markers to find additional markers for yield in the minicore and markers for grade, with the idea we could use these for marker-assisted backcrossing and selection to improve the grade for release of a, let's say, later second generation improved variety with tolerance to water deficit stress as well as improved grade. Finally, just acknowledge we're funded from several sources: USDA NIFA, the CARE and Hatch programs, Oleic Aquifer Initiative, National and Texas Peanut Producers Board, USAID under the Feed the Future Initiative, Peanut Mycotoxin Innovation Lab, and Peanut Innovation Lab. And mentioned that a number of people worked on this, both Texas A&M AgriLife research, for graduate students to contribute to this as part of their thesis work. And finally, acknowledge help from our associates at USDA ARS also.